Thank you.
Thank you very much, Katka. Thank you very much for this time for me. And yeah, it's true. I have a presentation about two topics. First part, it's focused on the metabolomics, the basic information about the metabolomics, sample preparation, and so on. And second part, it's the focused on our laboratory, and this part have one aim, and this aim is to show you the possibilities for cooperation with our laboratory. On this first slide, we have very known figure here. Maybe this area, it's very comfortable for you. It's the genomics and transcriptomics analysis. But for this day and for this presentation, we are here. We measured the small molecules. Typically, small molecules it's the molecules with the weights under the 1,000. Metabolomics it's the omics techniques, and he's separate on the three parts, because the small molecules it's a lot of type of the small molecules. First of them are lipids. For the measurement of the lipids, we have the special platforms named the lipidomics. For the measurement of the sugars, but sugars in this polymer form, we have the glycomics. And for the analysis of the amino acids, organic acids, and typically small molecules, we have the metabolomics. Okay, the metabolomics analysis is separate on the four pieces. First of them is the metabolomic extraction, because typically we have the sample for you. The typically sample is the leaf, and we need transport the metabolites from your sample, from the leaf, to the solvent, because on the mass spec we measure the solvents. Really depends on the type of the sample. I have three type of the sample. It's the butterflies, body fluids and soil extract or root extracts. It's the very easy matrix because our analytes are in the solvent. We don't need the extraction, the metabolites from the tissue or from the cells to the solvents. The second is the cell culture, and if we have the cell culture, depends on the type of the cell culture, because it's the big differences between the plant cells, because we have the cell wall, and it's the big problems for the extraction of the intracellular metabolites, because we need destroyed the cell wall. Typically, we use the freeze and tough procedure with the liquid nitrogen. For a lot of normal, I mean non-cell, non-plant sample, we typically used only the extraction to the organic solvents, and then we use the ultrasonic bath. The last, it's the extraction from the tissue. For you, it's the typical the extraction from the animal or from the leaves. In this tissue, we must very nice homogenize the sample, because the distribution of the metabolites it's different. Typically, we use the organic solvents for the stopping of the enzymatic activity, and in the second step we use the freeze and tough procedure, and then we continue with the sonification, centrifugation, and then LCMS analysis. Second step of the metabolomics analysis is the chromatography, because we have two possibilities for the measurement of the metabolomics. First of them, it's the use the mass spec, and second, it's the use the nuclear magnetic resonance. In our lab, we have only the mass specs. We haven't the magnetic resonance, but it's possible. But we have the different sample preparation. Chromatography. Do you know what is the chromatography? Because chromatography, it's work for the separation of the metabolites from the sample. Because on the start I have the extracts, and in the extracts it's include a lot of metabolites, thousands metabolites, and I need the separation during I need the separation during the time. And chromatography, typically LCMS is the liquid chromatography, or GCMS is gas chromatography. It's used for the separation of the metabolites. This is the typical outputs from the chromatography. This is the model chromatogram, and this is the real chromatogram. And we have two types of the chromatograms, because this is chromatograms from the total ion current. And if you look on the intensity, we have a really big intensity, and on the chromatograms we have, I don't know, 10, maybe 50 peaks. But it's not true, because in this chromatogram we have thousands peaks, but the peaks are inside in this curves. Because if we used the reconstructed chromatogram, that means I used the exact mass, and look on the intensity. We have E9 here, we have the E7, and these peaks are here, but it's invisible in the total ion current. Yeah, and here we have very nice peak for the sarcosine, alanine, and beta-alanine. If we know what I find. The chromatography information have two possibilities: quantitative and qualitative. Quantitative information is the peak area. We integrate the area under the curves, and this area and used for the calculation of the concentration. For quantitative information, we have the retention time. For each peaks, we have the two information, retention time, and we have the peak area. Each chromatogram have on the x-axis have the retention time in the minutes, and on the y-axis we have the relative abundance. Okay, here we have the same chromatogram. We have the sarcosine, alanine, and beta-alanine. And if we have the bad separation, we have the problem with the analysis. Because for the nice analysis of the alanine, we need nice separation of the alanine. Here is the methods how to calculate the concentration. Typically, we have two possibilities. We have the calibration curves methods, and we have the standard addition. But typically we use the calibration curves. We prepare the standard calibration solutions, and we construct the calibration curves. On the x-axis we have the concentration, and on the y-axis we have the peak area. And it's very easy, because we have the linear regression, and we have the slope, we have the intercept, and we measured the peak area in our unknown sample, or sample with the unknown concentration, and we calculate the concentration. It's very easy, but if you need the absolute quantitative information, you need the standards. It's impossible measure the absolute quantity without the standards. Next part is the mass spec analysis. Mass spec analysis, it's separate on the four parts. First, it's the source, because the mass spec, it measured only the ions, no negative molecules, only ions. And this is the reason why we need on the first step, we need to ionizate the sample. Second, it's the analyzer, it's for the separation of the molecules, and last part is the ion detection and data system for the evaluation of the data. Here we have the small information about the ion source ionization, because mass spec needs the ions, because during the mass spec, ions have the very complicated way. And if you have the ions, it's not problem curves the way of the ions. But if you have the ions, it's impossible use this special complicated way. We have three possibilities. We have the electrospray. It's the typical ionization techniques, but for the non-polar compounds, we use the atmospheric pressure photoionization on atmospheric pressure chemical ionization. But the principle is very easy. This is the end of the liquid chromatography, and here we have the the molecules without the charge. But we have on the ends of the needle we have the current, and we add the charge on the droplets. It's not ions because it's only droplets, and each droplet contains a lot of molecules. But this space, all this space, it's under big temperature, typically 350 degrees of Celsius, and we evaporate the solvents, and the space charge, it's the bigger and bigger, and after the Coulombic explosion, it's the special names. We prepare the analytes with the charge, and it's the ions. Typically, on the small molecules, we have charge 1. Okay, we have two possibilities. We have the positive ionization and negative ionization. Depends if we add the positive or negative current. If we have the negative ionization, we deprotonate the molecules, and if we have the positive ionization, we protonate the molecules. But this is very important because these two chromatograms are from the same samples. A lot of compounds have the signal in the positive and in the negative modes, but a lot of metabolites, it's impossible measured in the negative. Typically, the quaternary amines have the permanent charge, and it's impossible measured these compounds during the negative ionization. Yeah, because this is the chromatogram from the negative and from the positive ionization from the same samples. Mass analyzer. We have two possibilities. We have the mass analyzer with the low resolution and with the high resolution. For you, it's very easy because the instrument with the low resolution measure the mass only on 1 decimal place, but the machine with the high resolution analyzer measure the mass on the 4 decimal place. It's very important if I have the non-target metabolomics and I need the established the elemental composition. I need the high-res machine. Yeah, typically low resolution mass spectrometry, it's used for the quantitative for the quantitative analysis. Here we have the typically outputs from the mass spec. It's the mass spectrum. It's the graphs. On the y-axis we have the M ratio M and Z, but Z is the charge, and typically we have charge 1. And this is the reason why we have in the metabolomics on the y-axis only the mass, and on the y-axis we have the relative abundance. This is the mass spectrum from the HeLa cells, and we have for a lot of compounds we have only one mass. But what is the mass? because here we have the mass for the alanine. It's this mass, and alanine have this elemental composition. And if I need find the and prepare the reconstruction chromatogram and find the alanine, I must know the elemental composition and can calculate the mass. And the calculation of the mass is very easy because I have the 3 carbons, I have the 7 hydrogens, 1 nitrogens, and 2 oxygen, and it's the positive ionization, and I add one proton, and I calculate the mass. And if I have this mass, it's no problem. Analyze the alanine or different metabolites. Why we use the chromatography and mass spectrometry together? Because we have a lot of compounds with the same elemental composition. That means with the same mass. Because this is the same chromatogram. We have the sarcosine, alanine, beta alanine. But these 3 metabolites have absolutely the same elemental composition, but absolutely different biochemical pathway. And this is the reason why we need very nice separation. Because without the separation, I measure each these compounds together, and I haven't the information about the alanine. I have without the separation, I have the information about the sarcosine, alanine, and beta alanine. And this is the reason why we use the LC and MS combination, both techniques. Here we have the workflow for evaluation of the data, because here we have the chromatogram. This is the same. It's the sarcosine, alanine, beta alanine, and this is only the curves, but through it, we have in the curves, we have a lot of scans, because if we focus the retention time only on this second peak, this peak, we have a lot of scans for the 1 peaks, and in each one scans we have the mass spectrum. Yeah, and we need if I need the quantitative information, I need typically 15 scans per peaks. Okay, we have the full scan. I mean, I measured the molecule in the. If we go back, we have this is the full scan, because I measured the molecules without the fragmentation. But I have the possibilities for the fragmentation. And we used this fragmentation. We say MS analysis or MSN analysis, and we used this special type of the measurement for the identification of the compounds. Because if we have the bottle, is normal beer bottle, and I have the hammer, and I use the hammer on the bottle, I prepare a lot of pieces of the bottle, but in the mass spec, it's the same, because this is the alanine, and this is the part of the alanine. But in the normal, if I use the hammer on the bottle, I have different pieces. But in the mass spec, not, because I have the same hammer, I have the same energy, and I have the same molecule. I prepare the same pieces, and I used this fragmentation for the identification of the compounds. Okay, identification. Identification is a really bottleneck of the non-target analysis. I mean the non-target metabolomics analysis, because it's not problem. Measure the sample, and we use the software for the peak detection and so on. But I prepared a very nice table with thousands of the row, but I need the identification, and I have 3 possibilities. I have 3. This is the identification pyramids, and here we have the raw data, and we used only the mass for alanine. Yeah, because I calculate 90, and yeah, maybe this is the alanine, but maybe not. because if I haven't the retention information, it's the alanine or beta-alanine or sarcosine, I don't know. But if we have the MSMS spectrum, is the fragmentation spectrum, I have the next information, and the MSMS spectrum of the sarcosine and alanine is different, but it's not different if I compare the alanine and beta-alanine. And still, I haven't the information for the identification. Still, I have the information only for the annotation, and it's big differences between the identification and the annotation. And if I have the authentic standards, I measured the retention time, I measured the full scan and MSMS. I have the only in this is if I have this three information, I have identification of the compounds. And this is very important because if I need to identify the compounds, I need the authentic standards. And if I haven't the authentic standards, I haven't the identification, I have only annotation. And it's the really big differences. Why we have the problems if we have only the mass or MSMS spectrum? Because here this is the part of the glycolysis, and here we have the pentose phosphase pathway. And in these two biochemical pathway, it's very important the sugars and phosphorylated sugars. But big problems it's with the separation of the isomers, because here we have the ribulose, but the same have the ribulose, ribose, and xylulose 5-phosphates. But if I haven't the standards, I haven't any idea choose the correct peak only for the ribose. And the same is for glucose 6-phosphate and fructose 6-phosphates. It's the same elementary composition. Without the standards, I haven't the information what is glucose and what is fructose. And if I have this information from the biological interpretation, and I haven't the standards, it's impossible. Typically, it's impossible use this data for the biological interpretation, because here we have the mass, because mass spec measure the mass. Okay, it's not problem calculate the elementary composition from the mass, but here we have the three biggest database of the mass spec. Of the mass spectrum. We have the HemSpider, KEGG, and human metabolome database. But if we have, I don't know, the mass of glucose 6-phosphate, we have 194 matches in this database. This matches in the KEGG, and this matches in the human metabolome database. And it's impossible to say, yeah, I have the glucose 6-phosphates. I need the authentic standards. The last part is the data analysis, and here we are separate on the target and non-target analysis, because during the non-target analysis I measured everything, and during the data evaluation I find the interesting metabolites, typically after the statistic evaluation and so on. But during the target metabolites, I measured, I know what I want, because I measured only my interesting compounds or interesting metabolites. Typically, we start with the on-target metabolomics, and we found the interesting compounds for your topic or for your project. And in the second step, we used the target metabolomic analysis for the quantification of the compounds, because each analysis have the advantages and disadvantages. Because we have really big problems during the on-target metabolomics profiling. It's the identification and very bad sensitivity, because it's impossible set the mass spec exactly for the each compounds. But if I know the type of the compounds, I don't know, I need measure the alanine and amino acids, it's not problem set the mass spec for the special condition, but it's impossible during the on-target metabolomics. I added this slide because it's maybe important for you, because it's because I start with the metabolomics analysis from my side. I start with the sample preparation, but first step is the collection of the sample, and it's really important how I collect the sample, because if I have the problems during this part, it's impossible measure the sample. Because the first very interesting point is the standardization of collection conditions. Because typically I have different weather. It's a really big problems. I have big changes of the temperature of humidity. Maybe the biggest problem is the multiple people collect sample for one study, because it's a really big problem. Because typically, if I measured the sample from the study with these problems, we very nice separated the data based on the people, not on the treatment. And we during this process, we really need the standard operation process for the very correct information of the each people include in the study. The second interesting and important part is the stopping of the enzymatic activity. Because if I collect the leaves from the tree, and okay, but typically you use the plastic bag, and you add the leaves to the plastic bag and go to the next sampling point. But the enzymatic activity, it's still working in the sample, and depends on you. What do you want? What do you need? Because if you need the information exactly about this time point, if I harvest the leaf, you must stop the enzymatic activity. Typically, we used the organic solvents, or we used the liquid nitrogen. And next, it's the homogeneity of the sample, because it's a really big problems. Because if you, I don't know, we have 4 trees for the sampling, and one it's very near to the river, and second it's very far to the river. And typically we measured the very nice separation of the sample, but based on this, not on your experiments. And last is the blanks. Blanks mean samples mean the samples without your sample. Typically, it's the empty plastic bags. Yeah, because we need the information about the contamination. And if we haven't the blanks, we haven't this information, and a lot of peaks in your analysis. It's maybe contamination. It's not interesting for you. And typically, these peaks are very nice, very big, and perfect. But yeah, we need the blanks samples. Don't worry, it's really this is the shorter part of the presentation. It's the introduction to our lab. We are the part of the Institute of the Entomology. We are the Laboratory of Analytical Biochemistry and Metabolomics. This is our doors. Here we have the Institute of the Plant Molecular Biology, and we have the doorbell here. But our lab is focused on the mass specs. Everything in our lab we measured on the mass specs, and we have two labs. First of them is our LCMS labs, and second is the GCMS labs. Yeah, we separate on the chromatography, LC chromatography or GC chromatography. In the LCMS lab, we have a lot of platforms, because we say platforms, not the methods. But this is very these methods are very important for our maybe future cooperation. First is the target, non-target, and flux metabolomic analysis. We have in this time we have two platforms. First of them is the based on the high-leak separation. It's primarily focused on the analysis of the primary metabolites, not the secondary metabolites. And we have the second platforms based on the reverse phase separation. And this platform it's maybe interesting for you because it's based on the analysis of the secondary metabolites. For each metabolites, it's possible to measure the quantitative information. But our research is focused on the primary metabolism, not on the secondary metabolism. And this is typically the problems for the cooperation because we haven't the standards of the secondary metabolites. We cooperate with the Martin Wolf and Katka-san, and we have, I don't know, we have 100 standards of the secondary metabolites, but this is the number of the primary metabolites in our lab, and it's really big differences. Okay, next platform, it's our target and not target lipidomics analysis. It's based on the separation and analysis of the each classes of the lipids. Typically, in one sample, we found 350 lipids. Depends on the sample. The next is the analysis of the peptides, but typically we measure the small peptides. We have the border on the 50 units of the amino acids. Next is our analysis of the steroids, sterols, and bile acids. Next is the analysis of the vitamins, and now we established the new platform for the analysis of reactive organic species. That's mean typically the markers of the oxidative damaging of the cells. This is our equipment. We have two high resolution mass specs for the metabolomics. This is a low resolution, and this is low resolution for the quantitative analysis. In the GCMS lab, we have the analysis of the saccharides and polyols and the amino acids. In the GCMS lab, we must use the derivatization because it's impossible to measure the samples direct without the derivatization. Derivatization means change the structure. We typically use the functional carboxyl, hydroxyl groups, and we add the special part for the better volatility of the metabolites. In GCMS lab, it's possible to measure the quantitative information of the fatty acids, and in the both version, in the free fatty acids or the fatty acids, including in the lipids. The special analysis in the GCMS lab is the chiral analysis. It's the analysis for the separation D and L isoform of the compounds, typically of the amino acids on the organic acids. Maybe for you, it's very interesting, our volatile organic compounds platform. It's for the analysis of the volatile compounds. My colleague Petr Vodrážka had the presentation, I don't know, 1 or 2 months ago. I'm not sure about this analysis. And last is our sterols and tocopherols analysis. In the GCMS lab, we haven't the high resolution mass spec. We have only low resolution mass spec. Here is the small information about the sugar analysis, and maybe the interesting is this chromatogram, because during this GCMS sugar analysis we separate the sugars based on the mass. We have very nice recipe for the glycerol, tetrose, pentose, hexose, and disaccharides. This is the slide only for the volatile organic compounds analysis. It's possible measured the volatile compounds after the active or passive sampling, and in this time we measure typically the samples from the passive sampling, because we have the cooperation with the Martin and Katka, but we have the equipment for the active sampling. And this is it.